Hey guys, welcome back to my MCAT biology review. Today we're going to be going over the nervous system. I'm going to break this video up into a few parts, so this is going to be part one. Today we're going to be covering the action potential mostly, as well as how ion channels work and things like that. I would say this is pretty high yield stuff, and I'm going to try and make it quick. I also have two practice MCAT questions that I think will be helpful for you. Okay, let's start. So, when you're nervous, think energy. When you're dealing with the nervous system, you're going to be dealing with electricity, electrical signals, voltage, things like that. How does that work? I like to use the hot Cheetos and bananas analogy. It's kind of stupid, but it works. So to understand how the nervous system uses electricity, let's look at how cells work. So if you watch my other video, you'll know that cells have these membranes that are compartmentalizing. So this is the outside and this is the inside. In this analogy, let's say, there's a lot of these bananas inside the cell and only one outside. On the flip side, there's these hot Cheetos in the red on the outside a lot, but there's only a little on the inside. What do these represent? The red, the hot Cheetos. Hot Cheetos are very salty. This is sodium. Okay. Bananas, very healthy. The cell wants you to have them. Full of potassium, right? So you have a lot of sodium on the outside and a little on the inside. You have a lot of potassium on the inside and a little on the outside. Why does that matter? It matters because cells use the changing and the movement of ion concentrations across a membrane to generate electricity. So let me write that out because that's important. Movement of ions across a membrane generate electricity. How does that happen? Honestly, it's kind of complicated and you're not going to need to know it for the MCAT. So if you want to learn about it, you should learn about it, and you should look into something called the Nernst equation, and it has to deal with concentration gradients and electrical charge, things of that nature. It's kind of out of the scope of the MCAT. They're never going to ask you to calculate these things. So I wouldn't worry about it. Just pretend we're playing a game, and in that game, the movement of ions across a membrane generates electricity. It can be positive, it can be negative, and that's important because, as we know, neurons and nerve cells talk to each other with electricity, right? So let me show you. Here we have ion channels. Let's pretend this is the inside and this is the outside. Remember, on the inside, we have a lot of these potassiums with a little tip right here. And on the outside, we have a lot of these hot Cheetos, which is sodium, okay? And so we know that the cell membrane is impermeable to ions because it's nonpolar, it's uncharged. That's something really important you're going to need to know. We'll cover this more in a biochemistry lecture, but for now, this membrane, it's like plastic. You're not going to get ions moving through. If you think about a Gatorade bottle, you don't have water and the salts leaking through it. It's all contained. How does it get through? There are these things called ion channels. They're also going to be called channel pores, channel proteins, leak proteins, blah, blah, blah. The MCAT's going to throw a lot of words at you. Basically, they're proteins that make holes in the membrane that only allow specific ions or things to go through. So for example, let's call this one the potassium channel. This allows potassium to move down its concentration gradient. Okay, that means there's a lot of potassium here and there's not that much here. So if you have a ton here, every so often some of it's going to go out, right? And it's not as likely that it's going to come back through, although it could happen. At the same time, you have the same thing happening up here. You have a lot of these hot Cheetos. You poke a little hole into the hot Cheeto bag, all of a sudden, one or two spill out. These movements make an electrical charge. You can pretend that's lightning. So we see ion movements through the channels create a charge. Why is that important? Before we get specifically to action potentials though, let's take a little break and let's try an MCAT question. So, a recently classified epithelial cell membrane protein X may have effects on blood and cell glucose level. Figure 1. According to the data, what is the most likely substrate this protein acts on? This one seems kind of straightforward. I'm not too stressed about this. Epithelial cell, it probably doesn't matter. Membrane protein X, okay, it's going to work on the membrane. So it's a receptor or a channel or something like that. May have effects on blood and cell glucose level. Okay, so I'll keep my eye out for glucose levels. What's the most likely substrate this protein acts on? A substrate is exactly what it sounds like. It's just whatever the substance that the protein is working on. So basically it's asking what does the protein work on? And it's giving us some data. So let's look here. Figure 1, ion and glucose concentration inside and outside the cell. So it seems like it's not just giving us glucose, but also ions. Maybe that'll be important, I'm not sure. Plus and minus X. So it looks like they made a cell with it, and then they made a cell that doesn't express it, and then they looked inside and outside. So we have glucose, calcium, sodium, and potassium, and then here's the minus portion. In these types of things where it's straightforward like this, I think it's just good to compare. So in the glucose, in the positive, there's a high amount inside and a little outside. High amount inside, little outside. In the calcium, there's a little bit on the inside and a lot on the outside. But here, there's a lot on the inside and a little on the outside. So I'm going to make a note of this. That's one thing to look at. Next up, a lot of sodium on the inside and a little on the outside. A lot on the inside, a little on the outside. Potassium, a lot on the inside, a little on the outside. A lot on the inside, a little on the outside. When we don't have this protein, it looks like there's a lot more potassium inside the cell, which means this protein probably influences calcium. Because here when we have the protein, there's a lot more outside. Here, there's a lot more inside, meaning this protein, I don't know how it works, but it probably acts on calcium. So boom, calcium, easy. Remember though, you want to cover each single question. I've lost a lot of points on the practice questions when I didn't look at every single question and every single answer, because sometimes there's going to be one answer more right than the others. This one is pretty straightforward, but always look. Okay. So we remember that these ion channels, when you have cells that move through them, when you have ions that move through them, they generate electricity. I'm going to introduce you to something that you may or may not have seen before. It's called the action potential. And don't worry about it. It looks more complicated than it is. Action potential is a way that cells talk to each other. So you have a little neuron with its little axon. You have a neuron, little axon, neuron, little axon, etc. You get the idea. This is a spike in electricity. Okay? So this thing generates a spike and it moves down this big wire. Bzz. It shocks this, which generates a spike in electricity, moves down this wire, etc., etc. It's a way for these cells to send a signal. It's either on or it's off. Okay? It's analog. So what happens? We know that ion movement generates electricity. Here we see changes in electricity, so that tells us, okay, this is probably ion movement. Let's cover it. In 1, A neighboring cell delivers a shock through this action potential. Okay? 1, right there. Already covered. In 2, we see it goes from a negative to a positive. That is called depolarization. Okay? It's polarized because it's less than 0. Okay? When you go above 0, it becomes depolarized. At the same time, when you go towards the negative sign again, that's called repolarization. Okay? And if you go even more negative than before, that's called hyperpolarization. Okay, not too complicated. So we know the cell through some sort of ions becomes depolarized, becomes repolarized, hyperpolarized, and then flat again. How does that happen? It happens through ion movement. So it's weird to think that something so simple, just like potassium and sodium, can allow us to think, but that's how it works. We're basically these big biological machines that are powered by electricity. So, in 1, we get a neighboring cell that delivers a shock. Boom, it's shocked. Okay? In 2, we see all of a sudden it reaches this level and then it goes up. So what's happening? This is called, this dotted line right there is called the threshold. Okay. The threshold is like a trigger. If you ever reach the threshold, it's an automatic action potential. Okay. So anytime you're going from baseline to threshold, it's going to shoot an action potential because the threshold is what activates this ion channel. This is the sodium channel. So remember, we have a lot of sodium outside the cell. All of a sudden, it's going to rush in. Remember, ion changes across a membrane generate electricity. That movement is what's happening here. So when the sodium moves in, that is a positive change. But then we see all of a sudden it goes back down. What's happening? If we move to 3, at some point, let me draw this in a different color. At some point, at a certain voltage up here, this gets blocked, meaning sodium can't get through. So it's not going to create that positive voltage change. Instead, what's happening is potassium. Let me draw a little banana. Instead, what's happening is that the potassium is actually exiting the cell, which makes a negative change. Okay, so we have the threshold trigger that fires and it opens the sodium. Then at some point, the sodium closes and the potassium opens. It goes down, down, down. But what's happening here during the hyperpolarization? What happens there is that the potassium channels actually close slowly. Because they close slowly, they kind of overshoot. So they go down, 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 and right about here they start closing, but it's overshooting. So this creates something called hyperpolarization, which leads to something called the inhibitory phase, meaning because this is less than that negative 70 value, even if another cell shocks it at this point, it's not enough to get to the threshold because that difference is bigger. So maybe normally a shock would bring it up to here, but now if a cell was shocked right here, it would bring it up to there, meaning no threshold would be met, meaning no action potential would be fired. This is important for things like your muscle, for example. If you want to move your finger, you need an action potential and a certain movement in a certain way. Okay? If you didn't have this if you didn't have this hyperpolarization inhibition, you might overshoot the amount you want to move your finger, or you might move other digits. It would be sending an improper signal. So if I wanted to move my finger here, it might move here, and then I would have to correct. This allows it to be precise. One really important key for the MCAT is that this action potential will always look like this. Okay? It will never get higher. It will never get lower. It will never have weird things to it. They're going to try and trick you. Okay? Resist it. It's always going to look like this. Okay? Even if you get a lot of signals right here, it's still going to look the same. You're not going to get multiple spikes. It's always the same. Remember that. Almost done. So we saw hyperpolarization, okay? But how do we get back to that baseline? Because we know the sodium makes it go up, the potassium makes it go down, but then at a certain point we flatten out again, right? And at that point we're ready for the signal. This is through something called the NAK exchange, okay? The NAK exchanger, also known as the sodium-potassium ATPase, is a membrane protein that uses ATP to adjust the levels of sodium and potassium, okay? 3/4 of your cell's ATP for action potential cells goes to the sodium-potassium ATPase. This is a very important protein. What you need to know is that it pumps K in, okay? So during the action potential, remember, during that potassium part, as the potassium is lost and that graph is going down, we're losing potassium because it's going out of the cell, okay? But we need to get back to normal. So our friend right here, this monkey, the sodium-potassium ATPase, is going to, using power, grab that potassium. It's going to grab that potassium and it's going to bring it back in. At the same time, due to the nature of how this protein works, it exchanges that potassium for that sodium. So when it brings potassium in, it also has a mechanism that pumps sodium out. Okay? Why would this have to use ATP? Why do you think that is? It uses ATP because there's already a lot of potassium here and already a lot of sodium here. Even though we lost a little during the action potential, there's still about the same concentration. So to move something towards its high concentration, to go from low to high, it takes energy. There's already a lot of bananas here. The balloon is basically being blown, if that makes sense. It would make much more sense to energetically open the balloon up and air is going to rush out instead of blowing more air in, because that takes energy. So potassium, sodium, ATPase brings potassium in and sodium out. One last question, and that's it. Okay. SQA, a known snake venom, has been shown to covalently bind the NAV voltage-gated sodium channel ion pore. Which voltage potential graph is likely to be seen in the presence of SQA? There's a lot of words I don't know here, I'm going to be honest, but let's break it down. SQA, a known snake venom. SQA must be some sort of venom or protein or something, has been shown to covalently bind to the NAV voltage-gated sodium channel. Okay, let's draw this out. And this is something that I would do on the MCAT if I was confused, because you don't have a lot of time, but spending 10 seconds, 20 seconds to draw something out, if it means you're going to get the right answer, is worth it. So here's the NAV voltage-gated sodium channel. Voltage-gated sodium channel. Okay, that's important. Voltage-gated sodium channel. This means it's involved in the action potential. So normally, what the voltage-gated sodium channel does is it allows sodium to come in, right? Because there's a lot here and a little inside. SQA has been shown to covalently bind to the NAV sodium blah 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 ion pore. I don't really know what that means. Covalent bond means permanent bond in this case. Maybe it works like this. Maybe the SQA binds to the ion channel pore. Pore means kind of hole, so maybe it's going to bind inside this hole. And if that happened, that means that sodium can't get inside anymore. Right? It's just bouncing off. That means that that whole top part of the action potential wouldn't be happening, right? So let's look at the question. Which voltage potential graph is likely to be seen in the presence of SQA? So in A, that's just a normal graph. I don't think that's it. For B, we have no activation. This could be it. Let's look at the other ones. C, we have no hyperpolarization. That's due to potassium, so that's not it. And in D, we have no return. So we have the sodium action and it looks normal, but the potassium is blocked. So it's not this. It's not this. It's not this. It has to be B, right? It has to be B because none of the other ones are right, and we see none of the normal characteristic sodium up part. So if that sodium up part is blocked, it should just look like a flat line. And that's it. Boom. Here are a couple things I didn't cover, and you can learn more about this if you want. I don't think it's super high yield. There are two types of refractory periods, absolute and relative. So an absolute refractory period is one where you can never get a signal no matter how hard the stimulation of the action potential is. Relative means you can still get a signal, but you need a much stronger stimulus, okay? There's things as called spatial versus temporal summation. This refers to the initiation of an action potential. So are you getting a lot of signals to start an action potential in a similar time, or are you getting a lot of signals to start an action potential in a similar space? Okay. You might want to know that the sodium potassium ATPase is 2K in and 3Na out. It's powered by ATP. You can remember this by pumpkin to remember the direction of the potassium. So pump K in. And then finally, action potentials move down axons. And the thicker the axon, the faster it travels. So an axon that's really thick will move the electrical signal much faster than a thin one. And that is due to myelin, which is the basically substance that covers an axon, which makes it thicker or thinner. Okay, well, that's it for this part one. I hope this was helpful to you, and have a good day.